It is a pleasure to me to introduce Professor Michele Vacca, a highly regarded expert in the field of internal medicine with focus on hepatology, with a special focus on hepatic diseases related to obesity, including NAFL, MASLD, cardiovascular reactivity, and hepatocellular carcinoma. Professor Vacca is an associate professor at Clinica Medica Frugoni within the Interdisciplinary Department of Medicine at the University of Bari, Aldo Moro. He also serves as principal investigator at the Hepatic Metabolism and NAFL Laboratory at the Roger Williams Institute of Hepatology in United Kingdom, in partnerships also with the University of Bari. Professor Vacca's clinical and research interests lie in exploring the links between nutrition, lipotoxicity, and the complication of metabolic syndrome, such as hepatic steatosis, cardiovascular diseases, and related cancers. His ongoing research aims to uncover the metabolic pathway contributing to lipid toxicity and tissue damage in key metabolic organs. Utilize systems biology approaches, Professor Vacca seeks to identify biomarkers and therapeutic targets for treating metabolic diseases. His exceptional work has been part of prestigious projects like the Innovative Medicine Initiative 2 in the Litmus project, testing marker utility in steatohepatitis, and Horizon 2020 in the Elucidating Pathways of Steatohepatitis project, the EPOS, where he contributed with studies on steatohepatitis and lipid dynamics. Additionally, he holds editorial roles in renowned scientific journals, including BNC Nutrition and Metabolism and the International Journal of Molecular Science. Today, we have the privilege of hearing Professor Vacca speak on the important topic entitled How to Choose a Murine Model that Best Recapitulates Human MASLD, an unbiased ranking based on proximity to human diseases. So please join the meeting, enjoy the meeting. Professor Vacca, the audience is for you.
Thank you very much, Pablo, for the really nice introduction, and thank you very much to you all, including Claudio and Tanya, for thinking for me for this talk. Can you see my monitor, my screen, and the laser?
Yes, we see.
Okay, that's perfect. So as Pablo has introduced, the scope of my presentation is to try to direct our choices into murine models that best recapitulate human MASLD. I will present for this purpose an unbiased ranking based on proximity to human disease that we have recently published with the Litmus Consortium in Nature Metabolism. And you can find all the publicly available datasets published, because the scope of this project was to try to give a survey to the field to orient our decision into clinics. So the plan of the presentation is that I will present some key clinical and pathophysiological features of human MASLD that need to be replicated in preclinical models. We will therefore define together, in light of this information, the definition of an ideal preclinical MASLD model that we would like to see featured. And then I will present you an outline of the Litmus resource we recently published with regards to methods and the results. Together, we will draw some take-home messages and future perspective to then open up the discussion immediately after the talk. So let's start from the definition of steatotic liver disease. I will not invest too much time in this because I'm pretty sure that you are confident with this information. Into our labs, we use a biochemical definition, which is the intrahepatic triglyceride content. You basically do a simple Folch extraction fraction, and then you weight the lipid mass, and if triglycerides are more than 55 milligrams per gram of liver, you define steatotic liver disease. In clinic, we use an histological definition, such as the presence of lipid droplets more than 5% of the hepatocytes. But what we have seen over the last decades is that many different non-invasive prediction biomarkers and instrumental findings can really well correlate with the disease at the histological level. I'm not speaking about liver ultrasound, and because we know that it's not accurate and there is no correlation between steatosis evaluated at the ultrasound with any histological definition. The most accurate are represented by magnetic resonance imaging, for example, PDFF, when the fat fraction is more than 5%, the CAP probe with the FibroScan, and there are also some non-invasive biomarkers, like, for example, fatty liver index, that predict really well if the patient has fatty liver, and then we can plan some specific diagnostic pipelines for the patient. Saying steatotic liver disease does not necessarily mean speaking about MASLD. And over the last couple of years, we have seen many different changes into the name, from non-alcoholic steatohepatitis to metabolic disease-associated fatty liver disease, and then finally, probably, hopefully, the last one, which will be the metabolic dysfunction-associated steatotic liver disease, MASLD. Well, there's a change in paradigm with regards to the new definition and the new guidelines that have been recently published by Journal of Hepatology in 2024 and many other journals, and is the fact that if we identify a patient that either at biopsy or by imaging has steatotic liver disease, and there is the presence of any of the cardiometabolic criteria, such as overweight or obesity, change into glucose metabolism, increase the triglycerides, low HDL, or increase the blood pressure, namely the old definition of metabolic syndrome, we can define metabolic dysfunction-associated steatotic liver disease if the alcohol intake is below certain thresholds. And the change of the paradigm is not only in the way in which we address the definition and the diagnosis of the patient, but takes also in consideration the disease pathophysiology and the correlation of the disease with overweight, obesity, insulin resistance, and many features of metabolic syndrome. Epidemiologically, we know now really well that if the prevalence of MASLD into the westernized world is around 30%, when we look to patients which are overweight, obese, and or with type 2 diabetes, the prevalence can reach 50 to 100%. There is still a small percentage of patients that has a lean body mass index and no increase in waist circumference. We are speaking about 7%. However, as it will appear from the rest of the presentation, these patients are not metabolically completely healthy, and we need to get a little bit rid of the concept of overweight, obesity, and we should start looking more to adipose tissue dysfunction, as it will appear clear from the next slide. So it's really important to understand why there is this association and what is the pathophysiological connection between metabolic syndrome, overweight, and obesity, and the roots of hepatic fat accumulation into humans. With this purpose, different human studies using stable isotopes and also in the back in the days also radio-labeled isotopes clearly identified fat accumulation into the liver as resulting from the imbalance between hepatic lipid influx and hepatic lipid efflux. With regards to lipid influx, is important to point not so much to overweight obesity, but to adipose tissue function, and in specific, the dysfunction that leads to insulin resistance. 60% of the lipidome is coming from free fatty acids and lipoproteins that are coming from the periphery to the liver and accumulate into the liver. Why the epidemiological association with overweight obesity? Because what happens in overweight obesity and in insulin resistance, even if the patient is at a normal BMI, let's imagine, for example, lipodystrophic patients, is that the insulin cannot inhibit a physiological process, which is known as lipolysis, which is important to redirect the lipids from the adipose tissue to the liver during the fasting state. What happens when we eat is that insulin goes up, inhibits the hormone-sensible lipase and multiple other intracellular processes into the adipose tissue, therefore reducing free fatty acid release that cannot accumulate into the liver. If the adipose tissue becomes dysfunctional, free fatty acids will still be released into the postprandial phase, and especially if we are speaking about visceral adipose tissue, that whose venous blood goes directly into the portal vein, it will target directly the liver. Another route by which fat accumulates into the liver is represented by de novo lipogenesis, that in normal subjects contributes to 26% of the total lipid pool of the liver, but in patients which are adipose tissue insulin resistant and have hyperinsulinemia and have an intake of refined carbohydrates, particularly important with diet, we will have the activation of both the carbohydrate response element binding protein and the sterol response element binding protein that will push a physiological process that converts acetyl-CoA into free fatty acids that then are used to build up complex lipids such as triglycerides, phospholipids, and so on. So de novo lipogenesis is particularly important in patients with insulin resistance that are hyperinsulinemic, especially if the diet is not controlled. With regards to the dietary lipids, the contribution is really minimal. And the reason for this is represented by the fact that when we absorb lipids from the gut, they are not going into the portal vein, but via kilomicrons, they go into the systemic circulation, they release lipids into the adipose tissue, and the remnant kilomicrons have much less lipids compared to the originally produced ones. Therefore, the contribution is around 15%. Actually, there are studies that have shown that in high-fat diets, like in the westernized pattern of diet that we have in the US, in the UK, a little bit also in Italy is becoming prevalent, unfortunately, you will have that this contribution can go up to 40%. However, as I was discussing, the increasing influx not necessarily means fat accumulation, because our body is provided with two efflux systems, which are hepatic fatty acid oxidation and lipoprotein export, mainly VLDL production, that will get rid of the excess lipids. However, what happens into the hepatocytes of patients which are insulin resistant, especially when also hepatic insulin resistance develops? Well, we know that mitochondrial beta oxidation and the efficiency of the lipoprotein export goes down, and there is a negative correlation with body mass index. And as a result of this, the hepatocytes will try to counteract this suppressive mechanism by activating some extra mitochondrial oxidative processes, which are known as omega oxidation for very long fatty acids and peroxisomal oxidation, that have two issues. The first one is that from an energy perspective, they are less efficient. They produce less ATP. But on the other side, they also produce a lot of reactive oxygen species that will lead to lipotoxicity, apoptosis, compensatory hepatocyte proliferation, senescence that will worsen this vicious metabolic cycle, and also the production of multiple pro-inflammatory mediators that will prime the Kupffer cells to produce different cytokines, chemokines, and growth factors that will attract from the bloodstream additional inflammatory cell types. And the Kupffer cells will produce also a lot of TGF-beta ligands that will promote, together with the inflammation, the transdifferentiation of the hepatic stellar cells into myofibroblasts that from one side will sustain the pro-inflammatory milieu into the liver, and from the other side, that will be accumulating extracellular matrix, thus leading to the fibrosis that is one of the most important consequences of fatty liver if not controlled, if lipotoxicity is not controlled. So it's not surprising that the natural history of human MASLD implies that up to 40% of the subject with fatty liver will develop a pro-inflammatory and pro-fibrotic form of the disease that we call steatohepatitis, MASH, that has the potential to progress towards cirrhosis, hepatocellular carcinoma, liver failure, and death if the patient is not transplanted. Another thing that I would like to highlight, and this will be open for discussion after my talk, is that most of these patients don't die of advanced liver disease complications, but they die of cardiovascular disease, and this is a paradigm that, in my opinion, needs to be more implemented in terms of research because of the strong connection with the metabolic syndrome and with other cardiovascular risk factors. Another important thing that I would like to highlight, because this will be important to understand what are the features that we want to be replicated into the animal models, is that together with the definition of metabolic syndrome, different studies have identified the fact that the different dietary components can modulate the progression of the disease together with sedentary lifestyle. The polymorphisms in some genes which are involved in lipid metabolism, for example, PNPLA3 or TM6SF2, and of course there are also some other genetic influences like the gender, the females are protected, ethnicity. We know that Southern Asians can develop much more advanced stages of the disease at a lower BMI. There are, without any kind of doubt, some family clusters, and also the maternal life during pregnancy is really important in influencing the progression of the disease. So taking together all this information and considering that it's really difficult to put all these equations into the same murine model, what is the definition of an ideal preclinical MASLD model that we would like to have into our labs to test any kind of scientific hypothesis? We want, of course, to feature steatohepatitis with progressive fibrosis and hopefully hepatocellular carcinoma with regards to necroinflammation, and of course all the different histological components of MASH, such as steatosis, ballooning, inflammation, and fibrosis. We want the model to feature obesity and metabolic syndrome as main steatotic liver disease drivers. In this way, we can use these models to study complex systemic components of disease pathophysiology, for example, the contribution of the brain or the adipose tissue. And they can be used also to test anti-obesity drugs that are becoming increasingly popular and have effects which are outside the liver. We want the model to recapitulate intrahepatic molecular events occurring in humans, and I will show you today some data regarding transcriptomics done mainly by next-generation sequencing. We have also performed lipidomics in all our models. You will receive updates on this really soon. Another point is that most of these models can be really long in challenge, even more than 1 year. So we wanted to try to identify models that had a duration of the challenge that was acceptable for preclinical pipelines use, for example, for drug discovery. I have been speaking in plural all my time because this work was funded by the European Union and the European pharma industry. The project is an IMI2, as Pablo said. The name is Liver Investigation Testing Marker Utility in Steatohepatitis. It involved 54 different partners, including academia, pharma, and industrial, and one professional body, which is the YASL, 14 countries for clinical recruitment, and 47 million of investment. I have been involved into coordinating the preclinical animal platform, which is a custom phenotyping pipeline for in vitro MASLD models. The goal of the project was to establish a defined set of biomarkers that enable the detection of MASLD, the monitoring of disease progression, and the regression and therapeutic effectiveness, for example, into randomized clinical trials. However, while the activities were mainly based on humans, there was also this small preclinical group that has as a goal reverse translating human biomarkers. And this purpose was supposed to be relatively simple, but we discovered that it wasn't. And the main problem that we needed to face when we started 8 years ago this study was that the large variety of models that have been proposed to model in mice MASLD. To let you understand the degree of the problem, just within our consortium, more than 100 candidate MASLD models had been tested when we started, when accounting for species, genetic background, what type of genetically altered animals, time points, room temperature, or diet. And this is really a small proportion of what is available into the literature, up to a point that a recent meta-analysis compared almost 4,000 different study designs to model MASLD. Another big issue is represented by the nomenclature. There is not a consensus yet. So for the purpose of this presentation, we will distinguish the models as follows. We call high-fat diets models which are enriched in fat between 40 and 70%. They can come from different sources. They have really low cholesterol, below 0.2%. And in terms of carbohydrates, they are mainly represented by starches. This means that they have relatively low GI. The main mechanism of action of these diets is to induce obesity and adipose tissue dysfunction and free fatty acid release into the liver. However, they suppress de novo lipogenesis, and for this purpose, it has been proposed that modifying high-fat diets, the so-called Western diets or atherogenic models that have been initially developed to induce into genetically altered animals atherosclerosis, might have been really good also to induce MASH. They are still high-fat diets, 40-45%, either saturated fat or in the past trans fat. They are enriched in cholesterol between 0.2 and 2%, and the carbohydrates are topped up with refined sugar, either glucose, fructose, or the combination of the two. The Western diets still induce adipose tissue dysfunction, but they also induce, since they contain a lot of refined sugars, especially those that contain fructose, they also push de novo lipogenesis via carbohydrate response binding protein. At least theoretically, we will see that this is not necessarily the case. Again, to push de novo lipogenesis, another model has been developed, which is the so-called American lifestyle diets. And basically, they are either high-fat diets or Western diets supplemented with sugar water, either fructose alone, glucose alone, or the combination of the two. Another really large group of models that have been widely used in the past are the choline deficient models. And when I'm saying is a variety of models, is because you can find different concentration of amino acids, for example, methionine. And since these models induce body weight loss, it has been proposed that topping up fat alone or in combination with cholesterol or only cholesterol alone, you will have more damage and you will prevent body weight body weight loss, therefore making the models a little bit more meaningful. They have been widely used in the past because they are much quicker than the models that we have discussed before, and the way they work is to inhibit VLDL production because choline is important to build up phospholipids, and this means that you cannot build up efficiently lipoproteins without phosphatidylcholanes. Another model that has been proposed is the high fructose diets that are pushing mainly de novo lipogenesis by adding fructose into the diet or in the drinking water. Most recently, it has been proposed that also diets, like high-fat diets or Western diets, can be combined with chemicals, for example, carbon tetrachloride or streptozotocin, to specifically enhance some of the features of the models. For example, if you want more fibrosis or a diabetic background. So, having understood how complex is the availability of options and considering that we wanted to choose our best models to run biomarkers into these models, we decided instead of cherry picking to do a wide screening retrospective with regards to experiments that have been already performed, but we prospectively generated omics data to identify MASLD relevant murine models in comparison to human MASLD. With this purpose, we analyzed the data of 473 mice, either wild types or genetically altered mice, and 89 rats, fed with the different models that we have discussed before. In total, we compared 39 models plus carbon tetrachloride as a positive control for fibrosis. We collected information regarding phenotype, histology that was centrally stained and scored by the liver histology group, and gene expression data, mainly sequencing for a couple of models. We had macroarrays available. And we generated a dual ranking output that we called MASLD Human Proximity Score that ranks the models on the basis of metabolic significance or the ability to induce MASH fibrosis, and high ranking means closeness to the definition of ideal MASLD model. In terms of the quality of the data that are available, we used body weight, triglycerides, and cholesterol to model obesity and dyslipidemia. Unfortunately, we didn't have for all the models insulin and glucose, but we have performed into the supplementary tables a wide screening of the literature to show which of the models have a characterization on that side. This is a limitation of the study anyway. And then we looked to liver enlargement and damage, focusing on liver weight, body weight, AST and ALT, and of course, histology. With regards to histology, we still look to the canonical features of MASLD in humans, like steatosis, ballooning, lobar inflammation, myeloid bodies, and fibrosis. However, we don't use the human scores, but Pierre Bedossa and Dina Tiniakos developed an ad hoc ranking system, scoring system that had as a purpose comparing the murine change in histology to what is expected in humans. In other words, we want steatotic lesions to be pericentral and characterized by macro and or mediovesicular steatosis. If none of these features are present, the model gets a penalty. Balloon cells need to be clear. rounded, and eventually increased in size. Lobular inflammation needs to be characterized by small or medium-sized clusters, and F1 fibrosis needs to be only perisinusoidal or F2 plus fibrosis. In terms of gene expression, we started with the canonical approaches like differentially expressed genes and differentially regulated pathways. And then we used a recently developed tool named as a drug set enrichment analysis that is able to compare different data sets and rank them on the basis of proximity to a specific reference that in our case is represented by human data, by generating a normalized enrichment score. The higher is the ness, the higher is the closeness to the reference. Again, we have engineerized the pipeline to rank the models on the basis of the relevance for MASLD development or the relevance for MASH fibrosis. In terms of gene expression information that we have from the patients, we have used publicly available data sets from our groups and from collaborators, namely Professor Sanyal, my former boss, Professor Tony Vidal-Puig, and the EPOS consortium that was led by Quentin Anstey. We also contributed patients to this cohort, and we clustered the patients on the basis of the presence or absence of steatosis and progression of fibrosis. We didn't touch the argument of the ballooning and the old Bedossa definition of MASH because you know that at the moment this is disputed. And we focused on its differentially expressed genes or differentially regulated pathways that are either relevant for early MASLD development, meaning mild steatosis versus controls. And you will see that most of the pathways here are associated to metabolism, as you would expect. Pathways which are regulated at all the comparisons, so they are relevant not only for the development of MASLD, but also for the progression. And you will see a lot of wound healing happening here, mainly represented by inflammatory pathways. And pathways and its which are important for fibrosis progression. And you will see here, again, wound healing, mainly compensatory cell proliferation, cancerogenesis, stellar cell activation, and all the processes that lead to disease progression. So really meaningful and really robust because we were able to compare the same hits among the different human data sets. And we took only those that are highly replicable. We therefore focused on those pathways which are relevant for MASLD development or relevant for MASH fibrosis. Let's look at the murine data now, and let's focus first on the whole transcriptome. You will see that the color is not such red. This means that with an hypergeometric analysis, we have only a partial overlap around 50–60% of alignment between the direction of regulation of the murine transcriptome compared to humans. This may look a little bit depressing, but actually is really convincing because we are speaking about different species and mice are not little humans. Intriguingly, when you start focusing on disease-relevant differentially expressed genes in humans, as described in the previous slide, the alignment increases massively, and especially in the Western diets and in the choline-deficient models when supplemented with fat and/or cholesterol. The degree of alignment increases even more when you look to pathways, suggesting that the single gene is not such important, but the overall regulation of the pathway is really consistent to what is expected in humans. And you have a perfect alignment when it comes to all those genes and pathways which are important for all the disease stage and for disease progression. However, I would like to point your attention that even into high-fat diets and Western diets, the lipid metabolism-related pathways that are those characteristic of early disease stage are not such well aligned. And even worse is the situation when you look into the choline deficient models that also do not induce peripheral insulin resistance. And we will look into the details of the best models in a bit. So how you combine the data from transcriptomic with the other layers? We generated this MASLD human proximity score, and to help you to navigate to into this complex slide, you will find phenotype, histology, and next generation sequencing or arrays. You will find body weight gain in red, loss in blue, and you also find the information regarding fibrosis. The more are the stars, the more is the CRN score. Another important thing is that you find two rankings, the ranking for metabolic significance and the ranking for the ability to induce MASH fibrosis. And just to help you to navigate within the data, you also find a back-to-back comparison among the two rankings. Let's go to the data now, and let's focus to the hits which are important for metabolic significance. There is no discussion that Western diets alone or in combination with the sugar water, the so-called American lifestyle approach, induce MASLD in this metabolic context, either in wild-type mice, for example, black 6, or diamond mice, or in obese mice, for example, OB mice or melanocortin-4 receptor knockout mice. However, a proportion of these models fail in inducing MASH fibrosis. There are really good news that there is a subset of Western diets that also feature progressive MASH fibrosis when they contain 2% of cholesterol and or they are used in genetically altered animals featuring obesity, like for example the leptin deficient OB mice or the melanocortin-4 receptor knockout mice. And using the knockout mice also shortens the duration of the challenge, of course. What about the choline deficient models? Well, as expected, neither choline deficient models nor CD models supplemented with fat and or cholesterol have a good metabolic relevance. We have been widely using these models into the past because they quickly develop F2 fibrosis, but we need to keep in mind that systemically there's no metabolic syndrome in these models. The other intriguing thing and also surprising one is that these models did not offer a clear advantage on Western diets in mimicking human MASH fibrosis. So in light of these considerations, which kind of models we should use in everyday life, providing some good average between the needs to have a good metabolic model, but also of progressing MASH fibrosis? The best model is the Western diet 2% cholesterol Gubra amylin NASH model GAN, which is commercially available, and we have tested it into wild-type mice for 42 weeks and in OB mice for 12 and 23 weeks. Another good model is represented by a canonical 0.2% cholesterol Western diet, the canonical heterogenic one, when feeding obese mice. In specific, we have information regarding the melanocortin-4 receptor knockout mice for 21 weeks. But careful, because in wild-type mice, this model didn't develop F2 fibrosis, just F0, F1. Another model that seems to perform really well is the choline deficient, amino acid defined, high fat, high cholesterol 1% diet when feeding LDL receptor knockout mice for 8 weeks. Regarding these models, it has been characterized for the GAN and the heterogenic diet that induce insulin resistance, but careful that, as we discussed before, the choline deficient model reduces circulating glucose. So I would not test a semaglutide, for example, in this model. Let's look to the phenotype. All these models, with the exception of the choline deficient model, induces body weight. They induce hypercholesterolemia, liver weight body weight ratio increase, AST and ALT. All the models induce S3 fibrosis, a certain degree of ballooning and inflammation, and F2 fibrosis. However, I would like to point your attention to the fact that triglycerides are a little bit counterintuitive because they are either not significantly modulated, or sometimes also suppressed, even in the Western models. This is not surprising because, as we were discussing before, looking to the gene expression analysis, it's pretty clear that the metabolic pathways, not all of them, but part of them, are upside down into the mice compared to humans. And among those that are not really well represented, there are pathways connected to the de novo lipogenesis, for example, fatty acid elongation, PPR signaling, cholesterol synthesis, and glycolysis and gluconeogenesis. What I'm telling you here is that these models are yet to be perfect, and there is still space for improvement of the study designs to make them even closer to human. But there's no discussion that if your aim is to look to all the disease stage, fibrosis progression, and most of the metabolic pathways, they still do really well the job. Another thing that we wanted to understand is these best models can be used to study metabolic treatments in MASLD. For this purpose, we took the GAN model, another Western diet 2% of cholesterol, which is the amylin model, and a choline deficient high-fat diet, and we mimicked the treatment, for example, reversing the diet to chow, or doing caloric restriction, or treating with semaglutide. And what we found in all these models is that there is no discussion that there is an improvement in liver weight, body weight, AST, and ALT. So liver damage and fat accumulation seem to go down. This is confirmed also by histology, and also focusing on the human disease relevant hits that we have identified, gene expression and pathways that we have identified with the strategy that I described to you before. However, the choline deficient model, when placed back on child diet, increased body weight, triglycerides, and cholesterol as a result of reintroducing choline into the model. What I'm telling you here is that this model can be good, for example, to test an anti-inflammatory, anti-fibrotic protein, or pathway, but I would not use it for metabolic purposes because the results would be counterintuitive. Another important thing is that the best model, the GAN model, but also other Western diets at 2% of cholesterol, have been proven that they can spontaneously develop MASH hepatocellular carcinoma when fed for more than 48 weeks. So, I mean, they can be used also to model advanced stage of the disease. So, concluding, choline deficient diets do not adequately recapitulate MASH, but they may add value for specific purposes. For example, to test antifibrotic treatments, since they can feature advanced fibrosis in short time frames, below 12 weeks, and the translation can be partially improved by using rats and or perturbing cholesterol metabolism. For example, using genetically altered animals like the LDL receptor knockout mice or high cholesterol content. The Western diets are the most balanced options for most preclinical applications and ensure good translation to human MASLD, since they can induce metabolic impairment and progressive MASH and are close enough to human disease. Substantial fibrosis F2 can be achieved by combining a good balance between refined carbohydrates, better if you combine both fructose and glucose, and saturated fat, high cholesterol, and extended duration more than 40 weeks, or obese genetically altered backgrounds. So what is the direction that we will follow in the future, considering that our screening resulted into having not a single perfect model, is to improve the translatability of the murine models with regards to metabolic pathways. We will see into the last slide that there is chance for improvement of the study designs and the dietary formulas. It has been proposed that thermoneutrality can improve the metabolic phenotype. We tested these into the GAN model. We didn't achieve such good results, but in FDI, there are different papers that have shown it. There is a lot of discussion around humanized models, especially for lipoprotein metabolism, considering that the mice do not develop atherosclerosis. And we are seeing different large animals, for example, pigs, that may be more meaningful species, but there is a lot of discussion regarding costs and practicability of these kind of approaches, which is requires specific animal facilities. Another important thing is that the animal models, now that we have a subset that we know that perform generally well, need to be optimized to model more advanced disease stage, like cirrhosis, portal hypertension, and hepatocellular carcinoma. And as we were discussing into the first slides, also atherosclerosis. We didn't have the capability to look into gender differences because of the magnitude of the comparisons that we had. But of course, this is extremely important because we know that from a metabolic perspective and from a liver disease progression perspective, males and females do not behave the same. And there is an opportunity to optimize some ad hoc models for heterogeneous subtypes of human MASLD, like pediatric, lean MASLD, and or genetic variants like PNPLA3. Another point is that we are seeing a lot of in vitro options, like, for example, precision cut liver slices, organoids, organ on a chip, to model MASLD in a dish. And this is a provocative question to open a little bit the discussion. I'm not completely sure we are there yet, especially when it comes to modeling systemic metabolism, because you, for example, will use the interconnection between the gut and the liver, the adipose of the liver, the brain, and so on. What about how we can improve study designs and dietary formulas? The answer is really easy. We did a PLSD in our data set to try to understand what are the study designs and features of the study design that more correlate with our outcomes. And we found that single housing, dry environment, and some specific features of the formula drive mainly the disease. We should avoid very high fat diets, especially those low in proteins. We have a strong correlation of proteins with fibrosis. This also opens a lot of questions in humans, considering how high protein diets are becoming increasingly popular to induce body weight loss. We should favor formulas with palm oil, 1-2% and cholesterol, and combination of glucose and fructose in the formula, but not in the drinking water, because what happens when you put the calories into the drinking water is the mice will eat less pellet, thus leading to reduced damage. Please, before finishing, let me thank all the co-authors of this long journey that lasted 8 years, especially Yannis and Leah, that are my fellow co-first authors into this publication, and of course my former boss, Tony Vidal-Puig, that trusted me in bringing this to publication, despite I moved from Cambridge 4 years ago. This is my team at the Clinica Medica Frugoni of the University of Bari and the Roger Williams Institute of Hepatology, the founders, of course, thanks to the collaborators. And I would like to say that we are recruiting, we have open positions. If you're interested in our research, please get in contact. Thank you very much for your attention, and I would be more than pleased to take your questions.
Okay, thank you very much, Professor Michele.